Hour we heard a lot of administrative stuff, but we heard very little biomedical sciences, so why don't we just start with the introduction of the first module? Well, the first disappointment will be that I have some organizational points as well. There's only a few slides, there's some repetition in there, and then we start with the content of the first module. So I give a block overview. We've seen the overall program introduced by Willem Vonke. And then I give my introduction to the first module, some more specific details. And then we start with the first theme of the first block, which is predisposition in general. And I'll give some examples of what we mean by predisposition to diseases or disease risks. And then I will go in a little bit more detail on susceptibility or increased risk or predisposition to cancer. Actually, on the structure of block 1, I just have one slide, and these have more or less already, no, these you have not seen. One block has 4 modules. These modules are all 2 weeks. Every module has a separate coordinator. I'm the coordinator of the entire block, and also in Hassel it's Jean, and we are also the coordinator of the first module in which we focus on predispositions, particularly looking at the interactions of genes, because genes can predispose people to certain diseases, but there's also an interaction with environmental factors. Now, how that comes about, that's what we are addressing today. The second module is on gene regulation, particularly the research on gene regulation in relation to disease and disease risk. So we will have a closer look at, well, these genes that may also be linked to predisposition. How do they express? How is their expression regulated? You probably know already a lot about how you can measure gene expression, but how is it controlled? When does this control go wrong? And do these processes have something to do with the risk of development of certain diseases? Well, that's the theme of the second module, again 2 weeks. Then the third module is on oxidative stress and how oxidative stress can impact on cell signaling. Now, probably you all have heard about oxidative stress, and you know about the importance of antioxidants. Here we will go a little bit more in detail on what we actually mean by oxidative stress, how you can measure it. Probably also know a lot of that as well already, and how it's affecting effective signaling. What is the potential role of mitochondria in signaling or mis-signaling in cells that are suffering from oxidative stress? That's module 3. And then finally, the immunological mechanisms that play a key role in human health and disease. Well, our immune system is there primarily to defend us from certain factors that can attack our health, but we can also develop an oversensitive or hypersensitivity, or we can have autoimmune diseases that are, so where we have our immune system not attacking potentially harmful invaders of the biological system, but that we attack our own cellular or higher structures. So that's autoimmune diseases. These are the 4 modules, and today we start with the first one, predisposition. Some things about the schedule. The thing is, well, you can't read it. It's also not the intention that you read it, but we are right here now. It's 11 o'clock. See that? I'm talking to you, and everything that is indicated in yellow are the lectures that are organized here in Maastricht. There's cell study. It's already been indicated. It's important that you spend enough time. It's been emphasized. I can only emphasize again. A lot of time on preparing yourself for the journal clubs. Oh, sorry, that's too fast. The journal clubs are here on Friday, the first one. What is indicated here is an introduction to the library, That's only interesting for newcomers, so you can. There was an opportunity to subscribe to this this training. It's in the schedule. And there's 2 different sessions, also in Hasselt, which is in grey. So the lectures in Hasselt indicated in grey. These are the journal clubs in Hasselt, and the blue ones are the journal clubs and task assignments that are organized in Maastricht. In this schedule you still see 4 because we don't know in advance how many groups we will have. We will have 6 groups here in Maastricht, so in the final version of this schedule there's only 6 blocks. Yeah? The lectures in grey are the lectures in Hasselt. Yeah. So if you're studying here in Maastricht, you can have the bus 1 hour before departure, which means at 9 o'clock at the Kiss and Ride. And we need this form, we need to fill it out because we have to know, that's official regulations, we do have to know who is actually on the bus. I've been told this week, you may have noticed, it's the World Championships for biking. So there's a lot of extra bus traffic. And we only could arrange for this week a bus for 40 people. Now we are over 60. So we can have 60 in that bus, but some people will have to stand. So if you are, particularly for this week, if you have the possibility to go with your own car or otherwise to Hasselt, certainly for this week that would be the preferred option. Although there is a bus for 40 people to go. This is the second week, more or less the same structure. This is what Leo Köhler indicated in red. This is the introduction and the way you can subscribe for the electives. So that's in the late afternoon on next Monday. This is something new. After the first journal clubs, we will have a sort of a test exam. So just to let you see what type of questions can I expect and what type of open questions. We just practice with one of the questions that we had in the last examination, just to give you from the beginning an idea. This is the type of questions that you have to prepare yourselves for, because it helps you to study. Then you know what sort of level of detail is. It's typically the question about how much detail do we need to have, and that's I think that's very helpful. So it will give you a good idea about particularly the open questions. In Maastricht, we will have the task assignments on Thursday. The task assignments, in Hasselt will be on the Friday, and that's because of the majority of the tutors were not available on Friday for different reasons. And then on Friday there is this first introductory presentation of a research school. It has not been indicated which one yet, but we will let you know. Yes? In the afternoon? Yeah. It starts at 4, and this is planned until 4, I think. Oh no. Okay. I didn't know that. The only alternative we have is that we switch it to the morning. Okay, who is receiving a bachelor's diploma next Thursday? Okay, it looks like we have a serious issue here. I will look into it and see if I can change the rooms, locations, and the timing without some people dropping dead to the floor because it's so many times that we have a change in the schedule. Okay, that was yet administrative issues. This is more or less a summary of what Willem has already told me. We have topic lectures, so these are broader topics where we have concepts introduced. And in this module it means we have a lecture on predisposition. Actually it's my lecture that will start in 5 minutes. The focus an emphasis on cancer susceptibility. Complex diseases such as autoimmune diseases is a lecture from Pete Stevenson this afternoon. Dermatological diseases, fetal origins of disease later on. There are also research seminars on cardiovascular disease, liver and psychiatric disorders, which are more addressing scientific data. So it's less broader concepts, but it's focusing really on research data. So it's a different type of presentation, although every lecture has a little bit of both, of course. It's continuum. And then journal clubs and task assignments. We heard from Willem that it's student-centered, so it's the thing that you do. You have to prepare very well. It's collaborative learning. There is a tutor, but the tutor is not going to explain to you how the world works. You will do the talking, you will do the presenting. And if you're well prepared, and this is why we have all this issue about fun. Last year's coordinator stood here telling me, oh, this is fun and it's great if you prepare well. Well, I can tell you it's only fun if you do prepare well. Then you can contribute, you discuss. But if you come unprepared, it's not funny at all. Because you feel a little bit embarrassed. Am I asking stupid questions? There's nothing wrong with asking questions. If you have read the paper, if you have thought about it, and you really don't understand, then you probably are capable of posing the right question. I've read this paper and I can't figure out what the hypothesis is because it's not clearly stated. And if I look at the background description of the science, I come up with a different hypothesis that would be more logical. Now that may seem to you that you don't understand it, but it may be pretty much the case. We don't always select the best papers, which are papers also that may be a bit obscure or where you have data interpreted in such a way that it's actually not beyond any doubt. So things may be confusing, and if you are confused, it's not a silly question. It's actually one of the things that where we can see as tutors that you have been thinking about this data. So if you are actively involved, it can be fun. You know, it's not just fun, it's also contributing to a large percentage. These journal clubs and tasks make up in the first module 50% of your grades. So it's worthwhile doing this. You are actually working on your examination if you prepare for the journal clubs. Okay, that was all about organizational stuff. Now we're going to content. So if there's no more questions, I will not forget about this issue on Thursday. I will announce it at some point how we try to find to go around it. Let's now go to predisposition and what we mean by it. Predisposition, if you go just to Wikipedia, you see that it's indicated as the state of being predisposed or susceptible, I don't want to use that word, to something, especially disease or a health problem. So it means that you are susceptible at a higher risk of developing a disease. Actually, it's also indicated as a latent susceptibility that only comes about under certain conditions, for instance by a stressing factor. So it means that under certain conditions, one individual may be at the more increased risk as a result of a change. And the question is, of course, how does this come about? What are the culprits or the factors causing this? Well, it may be in your genes, so you may have inherited a predisposition or a susceptibility gene. It can also be something from the environment. If you grow up in a certain environment, you may also have this predisposition to acquiring a certain disease. It may be related to lifestyle. And often lifestyle is more or less influencing your environmental exposures, for instance. And we have a very specific environment, the fetal environment. So during pregnancy, so individuals that are not born yet are also developing a susceptibility to disease and how it works, we will get to that point also during this lecture. So it's a question, what was there already from the beginning, what's in an individual's nature, and what's nurture, what comes afterwards in your education, in your social environment, or in your physical environment. And this is something that's rather difficult to modify, and this is something that actually can be influenced, and is also then of course a good target or a good approach to improve human health or public health in general. Now these are the examples that we will have in this first module. I will talk about cancer today. The green lectures are the topics. The red ones are the research seminars. So we will have a whole range going from environmental, fetal origins, dermatology, autoimmune, liver, cardiovascular, and psychiatric diseases and their predispositions. Let's start off with the genes, the ones, the factor that we can affect the least. And well, the lighter side of relativity is this guy that says, and here's from his father, this is a terrible report. And then he says, well, it's not so bad considering the genes that you passed on to me. So it's sort of an excuse. Well, you gave me these genes, I can't help it that I don't perform any better. Well, it's the same with diseases. So if you get your genes from your parents, maybe there's nothing you can do about a certain susceptibility or a certain predisposition. Now, how do we know that? How can we find that out? Well, a lot of information comes, of course, from the Human Genome Project that was started back in 1990, and which tried to elucidate everything that we know about the genetic code. And just imagine that in one cell, of which we have trillions, you find 46 human chromosomes, 2 meters of DNA, building up 3 billion DNA subunits, being a DNA basis, and then a code approximately for 30,000 genes that make us who we are and respond us to external factors the way that we do. So if you have to maintain or use all this information, you can imagine that there's a lot of opportunities and possibilities where things can modify a little bit or can go wrong, which eventually increase your susceptibility to disease, or maybe just pass on a certain disease. So the Human Genome Project also resulted in a possibility to establish your personalized genome. And you can get your DNA sequence, it's not even that expensive anymore, and then you know the sequence of all your 30,000 genes. And it was initially estimated that 1,500 diseases may be related directly to disease, but now we think there's a lot more, and that it's a lot more complex. It's not just one gene, one disease. There's a lot of interactions going on, so we'll see later on. So this information you can use in many different ways. And you can use it to see if an individual is a carrier of a certain disease, because you have this specific gene, you are a carrier of the disease. An example is thalassemia, which is a defect in the synthesis of the hemoglobin protein, so it leads to anemia. And if you analyze your genes, you can identify carriers that have that may have this illness, so to say. You can also use it for pre-implantation or prenatal screening. So even if you're not born yet, you can test and evaluate. Even if you are doing in vitro fertilization, you can analyze before implanting the embryo into the uterus and select those that you know have a certain disease, like Down disease, because you then find trisomy, one additional chromosome, being certain that that would be a diseased individual. Now you can understand there's a lot of ethical considerations if you do this type of applications, but it's up there, it happens, and it's probably becoming more and more reality. Yeah, that's the disadvantage if you see this. You can use screening of newborns, PKU for instance, predictive genetic testing for certain diseases, like the breast cancer related genes, diagnostic testing for hereditary comatosis for instance, or even pharmacogenetic testing. You can establish if an individual has an improved or an impaired capability of metabolizing certain carcinogens, certain compounds, certain drugs. And in pharmacogenetic testing, you can see, for instance, for the cytochrome P450s, of which you know that they metabolize certain drugs, if you do that very rapidly or not. So, if you know your genome, you also know your future. Well, Affymetrix and Roche are seeking approval for new applications, particularly for these pharmacogenomics approaches, and it's becoming more and more reality. And we'll see in a couple of years that this is becoming closer and more applied, along with the ethical discussions that go with it. Now take this example, for instance, about drug metabolism. This is a lot of names. You have different types of drugs, proton pump inhibitors, lipid, beta blockers, antidepressants, that are chemically modified, metabolized by certain cytochrome P450 variants, and these can be polymorphic, which means they have different, small differences, which implies that they are working more rapid, less rapid. You have these very poor metabolizers here, indicated in red, for instance, or intermediate ones, extensive metabolizers, and even ultra metabolizers. They have a very high rate of metabolizing certain drugs. And if you then see, for this compound, for instance, if a normal dosage would be 100%, on average, it means that if you are a very poor metabolizer, you only need 25% of the dose to have the same effect. And if you have a very ultra metabolizing capacity, then you may need twice as much to get the same effect. Now, I don't know if you maybe know someone who uses this type of drugs. It usually takes very long before you have the right dosage to get the right response. And that's you need a certain time before you are at a certain level, so it can take a year before you have the right dosage for a certain compound. If you would know this information of an individual, you can make a much faster move towards the right compound. Now this is just one type of compound. You can also see that sometimes it doesn't make that much of an effect. But just something as simple as paracetamol. If you have an operation, everybody gets the same dose. Well, just take two. We know now that paracetamol, if it would have been evaluated again, it would not be allowed on the market because of its hepatotoxicity. We know at very low dose it's an extremely hepatotoxic drug. Never say, oh, just take a paracetamol, because it's really damaging your liver. And yet we know that there's also a lot of inter-individual differences how people respond and metabolize a simple thing as a paracetamol. And we also know that the level of liver damage after using an overdose can be depending very strongly on your metabolic status. So there may be ethical issues on the one side. There are also huge benefits of using this information in pharmacotherapy. Now these were just cytochrome P450s, one gene, but one gene is not always linked to one disease. So this is more or less the simple way. If you have an autosomal dominant trait or characteristics, then you get Mendelian. If you look at how these pass on to the next generation, they follow Mendelian statistics. But that's only for monogenic things. So if you inherit one gene trait, passes on this characteristic to the next generation, or if you have a defect gene, you inherit the disease. But usually, or in many cases, these diseases are much more complex, and you have interactions between different genes, or between genes and environmental factors, a bit like the gene metabolism of drugs. And so then the inheritance of a mutated gene doesn't necessarily predict the disease outcome or the susceptibility or the severity of a disease, because it depends. You can have this gene, but if you don't have, you may have a compensating gene that takes over this job. So then you need to have two specific mutations in two or more genes. And so that's something that you have to look into. So there's more or less a continuum. You have major genes that affect the phenotype, which is high or low risk. Modifier genes that can compensate a little bit. You have environmental factors, which are non-genetic at all. And so here you see some diseases that are very strongly influenced by your genotype, like Huntington, cystic fibrosis. These are two colon-related diseases, hereditary non-polyposis, colorectal cancer, schizophrenia, etc. And you have, of course, outcomes, particularly lung cancer is, of course, determined by environmental factors like what? Smoking. Now, car accident is probably also not that much genetic related, so it's usually more complex. A monogenic recessive disease, cystic fibrosis. Cystic fibrosis, in proper English, Thaislijmziekte, is related to a mutation in a gene, the cystic fibrosis transmembrane conductance regulator. It means that you produce mucus, which is it's more sticky, it's more difficult to get rid of, and it has something to do with the translocation of chloride ions, and it regulates more or less the osmotic value of the mucus. And you can imagine if there's something wrong, that the fluidity of the mucus is decreased, and that's what happens, and it can strongly impact on pancreatic function, so insufficiency, airway obstruction because of this very tough mucus, and infertility as well. Now, if you look here, because we know there's a mutation in one gene involved, it still has a lot of environmental factors and modifier genes, in addition to this specific known gene that has different allelic variations, so there are different forms of it. If you have these forms, we know that this pancreatic insufficiency occurs in 90% of the patients, but not all. See that in most male cystic fibrosis patients, you see that fertility is clearly affected. But in regard to the pulmonary disease, that's very variable, and that's also had something to do, of course, again with environmental factors, the risk of infections, the use of antibiotics. If you use your medication in time, you can modulate that part of the risk. Or tobacco use, which also affects the severity and frequency of pulmonary disease and cystic fibrosis. So even if you identify one key gene, there's modifier genes modifying environmental factors. So that was one, that was genes. And we already said something about genes and environmental interactions. Okay, these are two different flowers, you would say, but genetically they are still the same, and that's because they grow in different environments. So these blue, you can buy one of these bloom hortensias, in a flower shop, and then you put them in your garden, and then the next year they turn up to be this dreadful pinkish color, which I personally hate. So there goes my garden, and it's ruining. This has something to do with the availability of iron. And so if you have a different environment, it helps. If you just have some rusty nails put around the roots, then you get a blue one. It looks like it's something different, but it's just a phenotype. Its appearance genetically is the same plant. Oh, so it goes for flamingos. If they feed differently, they look quite different, but it's actually the same. It could be the same animal. All sorts of organisms. These are water fleas. And in presence of certain pollutants or even predators, they get a completely different phenotype. They form these helmets that can help them to protect themselves to a certain extent from certain predators. So they look different, but the genotype is really the same. So pollution, presence, presence of predators, whatever it is, can modify the phenotype. Now, what sort of pollutants do you think in main categories could be of major influence on human health and human disease? Shout something. Sorry? Particle matter. Oh yeah, I gave that lecture in block 4 years ago for you. Yes. Ambient particulate matter. What else?
UV.
We've been telling this is participatory education, student center, we want you to respond.
Ultraviolet light.
Ultraviolet light, yes, you can see, well, you could see the sun if it wasn't so. Ultraviolet light, yes. What else? Radiation. What sort of radiation? Gamma radiation, ionizing radiation, ultraviolet, okay. More?
Heavy metals.
Heavy metals, very toxic. More. So we heard some chemicals. We heard some physical factors, radiation. That's not a chemical, unless it's alpha emitters. Other type of major categories?
Bacteria.
Bacteria. Okay, biological factors. Do we know more biological factors than bacteria? I heard something.
Maybe.
Fungi. Oh yeah, fungi. Yeah. Yeah. Fungi can produce chemicals, but they can also produce spores that can give allergic reactions. The flu, is that a bacterium? The virus? We're almost complete. We had some chemical factors, biological, physical factors. We also have social and psychological factors. So we heard some examples here, bacteria, viruses, radiation. Temperature and noise also fit into the physical factors. But this is something that I've not heard. The psychological, cultural background, social pressure, stress are often related to lifestyle. And they are maybe the strongest ones. So it's very difficult to escape from a beer if you're... Sorry? Oh, it definitely is. I'll show you why. Smoking is something that you do under social pressure. Nobody starts smoking in a social context. Occupational exposures. We have these signs on our laboratories. Lab coat obligatory. Now, you don't want to know what happens and how often it happens, but this is something that is also determined by your social environment, something that's a habit. So social pressure can lead to situations that are very much determining your predisposition to disease. And now, yeah, and we are sort of laughing about this, but it's actually really serious. The World Health Organization stated that as much as 24% of global disease burden is caused by environmental exposure, so it can be averted quite easily. If you don't smoke, the risk that you get lung cancer decreases by a large proportion, more than 33%, even if it's children under the age of 5, and they usually involve heart, lung, and bowel disease. So let's look a little bit further in this lifestyle. This is not obvious. This is also lifestyle. Why is this lifestyle? This young guy is predisposed to obesity, not because of his genes, but because this may be his father or his uncle or somebody else he looks up to, and that's what he wants to be. So he wants to wrestle with him and get him to the ground. Now that's also environmental predisposition that has nothing to do with genes but with social background. This is another example. Pima Indians, they live in Arizona. And there used to be nothing wrong with these people, let's say a century ago. But nowadays they have the highest incidence of diabetes in the world. Why is that? Because their cultural and social environment has drastically changed from an active way of living, doing different things, having different food habits, dietary habits, to something that is really disease-burden, predisposes them to illnesses. Now, where does this come from, this obesity? Is it a tendency to overeat yourself, gluttony, or is it just laziness? Well, it's, of course, a bit of both. You can see here in, this is a percentage for all time points. Over time, you see that the energy intake has gone up, but also has gone down. And you see still that the percentage of obese people increase after the 70s. So this cannot explain why the number of obese people is increasing. If you look at the number of cars per household or television viewing hours per week, then you see that if you change the way you move around, if you used to do this by car, on foot, or by bike, or whatever, on horseback riding, or by car, it makes you have a different energy expenditure. So it's energy intake in combination with energy expenditure. And you also see that an increase of the body mass index, if you follow this over the years, then you see that the same areas, particularly in the south and the east part of the United States, go along hand in hand with increased diabetes. This is the final slide on lifestyle. If you look at these figures, you see that a poor diet, physical inactivity, contributes to a large amount in total deaths in the USA. But also tobacco use and alcohol consumption. If you add up all of these and compare it to other external environmental factors, then you see that these things that we usually make fun about, smoking, alcohol, eating too much, things that we like to do and can easily avert. It's really easy. If you don't want to smoke, you don't smoke. If you don't start to smoke, or you drink moderately, or you don't drink at all. That's where the major health aim at public health level can be gained. And we can try to reduce toxic agent in our ambient air, or we can try to reduce microbial contamination of foods. But the easy, what seems to be easy and is under social pressure mostly, or as coming from a cultural background, is the impact of looking at that. And those factors is actually highly important. Okay, then something about the fetal environment, which is a very specific one, and Leo Köhler is going to have a full presentation on that next Monday. Again, smoking, alcohol, or a healthy diet, or malnutrition, they all determine how young individuals develop in early, it's not pre-life, in early life before being born. And well, again, smoking, because it's a very typical thing that we all know that there's a lot of conclusive evidence that infertility, central problems, low birth weight, prematurity, and sudden instant death syndrome, reduced lung function, can be the consequence of smoking during pregnancy. There's also substantial evidence for other things like ectopic pregnancy, so pregnancies outside of the uterus, respiratory symptoms, and indications for all sorts of other diseases as a consequence of smoking. Here you see this is smoking in a household, so it's not necessarily active smoking of the pregnant mother, and the increase of these sudden instant deaths compared to the cigarette smoke today. So this is quite convincing graph. Now how can that happen? There's different ways that it can be caused by nicotine, that is, has a constrictive compound, so the blood, the child under development can have impaired oxygen or nutrient supply. Can be displacement of oxygen by carbon monoxide, because we know that carbon monoxide is one of the compounds in cigarette smoke. It can impair vascularization, or it can be direct toxic effects of heavy metals that were also implicated. It has a direct impact on the development of the fetus. So fetal origin of adult disease, so low birth weight, is also associated with diseases that usually occur in later life. It's not necessarily something that is related to low birth, so something that you can measure or establish immediately, but hypertension, insulin-resistant diabetes, or metabolic syndrome, they occur in later life. So something is happening already in early life development as a result of environmental exposures that has a big impact on later life. Actually, some of these effects pass on to even the next generation, and that's what we call metabolic or genetic imprinting. So it's adaptive responses of the organism to specific nutrient conditions in early life. That is not necessarily a change in the genetic code, but can also be passed on as a consequence of epigenetic changes. So changes in the DNA methylation that result in modification of gene expression. Often dose response related, or sometimes you need a certain threshold, and it has to occur in a very specific time window during the development, so a critical window, intergenic window, where the development of the fetus is very sensitive for this. Okay, Leo Carolo will tell you more about how that functions. Okay, that's what I wanted to tell you about predisposition in general, the role of, I think I emphasized it sufficiently, of genetic factors being key, but also the interaction between genes and environment. So now let's have a further look into the predisposition to the development of cancer. Probably you all know what cancer is. I can't imagine that you don't, but just to be sure, what is cancer is a class of diseases in which a group of cells display uncontrolled cell growth, invasion, and sometimes metastasis, so spreading to other locations in the human body. That's what cancer is. Now, how does that come about? You have initially normal cell division. Something goes wrong during the transcription, or a cell is modified at a critical point. You have cellular damage, and in many occasions this is very efficiently repaired. We are really used to being damaged. So that was initially the idea. If you have exposed to one carcinogenic compound molecule, that I'm pretty sure that's not true. You have a very good DNA repair system. But if it's unrepaired, then there's a second line of defense. If its modification is lethal in itself, the cell simply dies, or if this damage is so extensive, the cell kills itself by a process called apoptosis. So then nothing goes wrong. But if this one nasty cell escapes all these defense systems and starts dividing, and accumulates maybe another mutation or a second or a third, then an uncontrolled cell division can be really out of control and result in a tumor. And this is a summary of the hallmarks, so the key points during the carcinogenic process. So normally we start with self-sufficiency in growth signals. So if you have the control of the growth systems in your cell, then there's nothing wrong. But if you continue to stimulate your own growth signals of a specific cell, then the cell can start dividing. Or you can, certainly in combination with an insensitivity to anti-growth signals, start to proliferate. If something like that has gone wrong and your defenses, like the apoptotic process, is evaded, so you pass by this other mechanism of defense, then the problem only gets worse. The formation of blood vessels, of course, if a transformed cell is invading a certain tissue, or the tissues maybe, at a distant place than where its original formation took place, has sustained angiogenesis, limited replication potential, then you start to build up. So these are the main 6 hallmarks of the carcinogenic process. Now let's look at cancer risk or cancer development as an environmental disease. We know it's an environmental disease. We know that there are certain chemicals that are carcinogenic. We know that cigarette smoke contains a lot of these compounds. We know that radiation, gamma radiation, alpha emitters, all sorts of different types of radiation can modify the genes, make DNA mutations, or some viruses or bacteria can even have a carcinogenic potential. So cancer can be considered as an environmental disease. Numerous compounds in cigarette smoke are known to change the genetic code. We know that sun and radiation, one of the examples that was just mentioned, can increase the risk of skin cancer. If you live somewhere near the North or South Pole, then your risk of skin cancer is relatively low as compared to if you are living or have the habit of exposing yourself a lot to UV radiation. And we also know that in different regions of the world, we find differences in incidences, so the occurrence of specific forms of cancer that are quite different. So for instance, in Japan, we see a lot of stomach cancer, which is related to the consumption of who knows what. Anybody know?
Mercury.
No, not mercury, it's salt. It's a very salty diet. So that's the main risk factor in Japan. China has a high liver cancer incidence, partially related to a fungi product, aflatoxin. So that's why there's a high incidence of liver cancer in China, also other Asian countries. Skin cancer in Australia, well, that's obvious. Some cervical cancer, lung cancer. So there's different major types of cancer in different parts of the world. Now, if you look what happens, taking this Japanese population, looking at the first generation migrants, or the second generation migrants migrating from Japan to Hawaii, what you see here for stomach cancer is that in this first, second generation, the incidence of stomach cancer decreases and comes closer to the cancer rate of the population in the area where they live. The other way around, for breast cancer, which is traditionally very low in Japan, it increases towards what we see in Hawaii. So these people have not changed genetically, so it must be an environmental factor. So this more or less confirms that cancer is an environmental disease. We can also look at from the other side, cancer as a genetic disease. We are these factors again, but we know that these compounds or viruses are only carcinogenic because they interfere with your genome. So you have inherited certain genes, and if you have specific modifications at certain sites where your cell regulation or proliferation is regulated, then you can find these modifications. There are also inherited conditions that increase risk for cancer. So this whole list here: retinoblastoma, xeroderma pigmentosum. I will come back to that one. It's an enormous increase in cancer risk. There are certainly, this one was already in the previous slide, familial adenomatous polyposis, so people that develop intermittently a large number of benign tumors in the colon that eventually have a high risk, if they are not removed, to develop into colorectal carcinoma. So there's also inherited conditions. So in that sense, you can also say that cancer is a genetic disease. Now, let's go further and look what may be behind all of that. There's two classes of genes that we discriminate: oncogenes and tumor suppressor genes, where these oncogenes stimulate cell survival and proliferation and have to be activated. So these are actually quite normal genes that are involved in the regulation of cell division. If they are activated, then they can result in uncontrolled cell growth. Or tumor suppressors that inhibit cell survival and proliferation and have to be, so that's also a normal mechanism, and if they are inactivated, then you lose this suppressive activity of these genes, and then they also increase the risk of cancer. Now, starting first with the oncogenes. It has been known that viruses can cause cancer, and certain viruses transform cells by inserting a piece of viral DNA into the cell, and these are indicated as oncogenes. So it's a bit of genetic information that can stimulate cell growth that comes from outside of the normal human cell and transforms a normal cell into a cancer cell. But we also know that normal cells contain these non-cancer causing genes that are closely related to these viral types of oncogenes, because otherwise they could never perform such an action, of course. And these are called proto-oncogenes. They are like these oncogenes, but they are proto because they are not yet oncogenes. They have to be activated. And then later it was demonstrated that a variety of tumor cells contain oncogenes which are transformed, which inform normal cells growing into culture to cancer cells. So these proto-oncogenes is the normal state that has to be activated in order to transform a normal cell to a cancer cell. So we then discriminate between the viral oncogene, which is brought in by the virus, or a cellular oncogene, which is a normal part of our genome and has been activated by changing it from a proto-oncogene to an oncogene. So it's gain of function. This is a normal proto-oncogene. If you have UV light, a chemical that is carcinogenic, it modifies it into an oncogene, and then it starts to behave like cellular, like an undividing cancer cell. So these are some examples, functions of oncogenes. By affecting cell proliferation, RAS gene or RAF are examples, EGF, so they stimulate cell proliferation. Cell differentiation, some other genes, so they can maintain the cell in the proliferative state. Or cell survival, which activate BCL2, which inhibits apoptosis and maintains cell survival. So once the cell can be transformed, they keep the cell in a surviving state, which then contributes to cancer development. Now then a look at the tumor suppressor genes. If you have a normal cell, these genes prevent cancer development, but if you activate, or let's say inactivate rather, or remove or inactivate these genes, then you get a cell that is damaged. But in this case, if you have one of the pairs, so you have two copies of this gene, if you have still one gene functioning correctly, then you still can have this suppressive effect. So here you need both of the genes to be damaged in order to have the effect of tumor suppressor gene being inhibited. And so gene functions can inhibit cell proliferation or development at different levels, for instance at the level of the receptor of certain growth factor, the signaling proteins, or at the level of the transcription factors. So that's always inhibition of at one of these levels. So in contrast to the oncogenes, they must be inactivated, means both copies must be lost. So if you look here at a normal cell, you have a first mutation event that inactivates the tumor suppressor gene, but then you still don't have the unfavorable phenotype, so to say. You need a second mutation event, and then you get this stimulation of cell proliferation. Now also here you can have a genetic factor, so if you inherit one copy of a defective tumor suppressor gene, so which is indicated in this one, and so for instance for retinoblastoma, if you inherit such a genetic trait, there is no increased tumor risk. But of course the chance that you get one specific hit in the other copy of the gene in one of these trillions of cells, then the likelihood that that occurs is much higher. And so occasionally, if you have inherited this genotype, then in this copy you can have excessive cell proliferation leading to retinoblastoma, and that occurs, of course, the likelihood that that occurs is much higher because you already have in all of your genes, this single mutated copy. So tumor suppressor genes inhibit oncogenes, suppress proliferation, induce cell death, or repair, or inhibit DNA repair, so the prevention of a mutation. So they are loss of function, recessive mutations, not dominant, responsible for hereditary forms of cancer. And if you are heterozygous, then you will require just one mutation. So it's all about statistics, it's chance calculation. So the risk increases, of course, with age, because if you have a longer time of exposure, a longer time that things can go wrong, and the incidence of cancer risk of aging increases because the time that you have to accumulate these mutations increases. It's actually quite logical if you understand the process behind it. One additional example is if you inherit a defect in DNA repair. Who's familiar with this disease, xeroderma pigmentosa? Have you heard of it? Okay, this is a very serious condition. It's an autosomal recessive genetic defect, and it's a reduced or elimination of nucleotide excision repair. So this is a repair system that can identify modulated DNA bases, which are, for instance, modified chemically by a chemical carcinogen. It's recognized, it should be removed, or it's induced by UV light. You have these two thymidine DNA bases that are linked together. And this nucleotide excision repair normally identifies this genetic problem and is fixing it. But is completely eliminated or reduced. And if you have such a mutation in a tumor suppressor gene like P53 or in oncogenes, this may result in cancer risk. So you can understand if you have that in every cell because you inherited this defect. If you are exposed to UV light, already patients very young, that are very young, develop skin cancer if they are exposed for a very short period of time to UV. so they can never leave their homes unprotected, so they have to cover gloves, they have to cover their faces, they have these masks on in order to prevent DNA mutations as a consequence of UV radiation, and otherwise they end up with... This is actually one of the nicer pictures. If you want to see what can happen really, then you Google on this and then you see that really young children already have multiple tumors on their face or hands when they are exposed to UV light. So this emphasizes what I already said. We are damaged continuously by chemicals, by radiation, by radiation, cosmic radiation. And we have developed a system of DNA repair that is very efficient. And if you go out on a day like yesterday, you get millions of DNA mutations. And you can measure them in urine as a consequence of this nucleotide excision repair. So it's an environmental factor that sort of pulls the trigger, but we have defense systems. If a defect in your repair system, like this excision repair, is in every cell, then your risk of cancer really increases. This is the last example. That's the polymorphism occurring in enzymes that metabolize potentially carcinogenic compounds. Let's take, for instance, benzoylpyrene, a polycyclic aromatic hydrocarbon that you find in cigarette smoke, but also in grilled hamburgers. Well, not professionally grilled ones, but the ones that I produce, looking black, etc. These compounds, they are known as carcinogens, but they only modify the DNA by forming DNA adducts if they are metabolized. So they have to be, in order to get rid of these compounds, they need to be hydroxylated. So there's oxidative metabolism involved. That's something that is done by different types of enzymes. And during this elimination process, they are very shortly activated into reactive intermediates that can bind to the DNA, and then they form so-called DNA adducts. And so they damage the DNA, and then afterwards they are removed. So we have different polymorphisms, so again, genetic variants of the same enzyme that can produce more or less active forms of this metabolizing capacity. And if you look at this DNA damage in lung cells in relation with smoking, then there's not really a very good correlation. You say, well, it's a little bit lower here, but if you cut off here, there's not really a good correlation. But if you look at the different genotypes, so there's a high, medium, and a low metabolizer variant, genetic variant, so heterozygous in a genotype of an enzyme called NPO, then you see that it makes a lot more sense. So the high metabolizers, so that produce in a short period of time a lot of these reactive intermediates that can bind to the DNA, indeed you see that there is an increased level of DNA damage. And the low metabolizers who do this at a very low pace, and these intermediates are also removed in what we call a second phase elimination process, detoxification. And you see that the level of DNA damage is much lower. And so you can imagine that if you have this genotype of the polymorphism, you are more susceptible than when you have another genotype. Now, and this is if you look at cancer epidemiology, so you look at cancer risk in relation to exposures in larger populations, that you see often very confusing results. At one point you read this is very bad for you, and then another study in another population does not show a clear correlation. And that may be just because these epidemiological studies usually don't have molecular biomarkers. So if you ignore the genes, then you get this plot and you think, well, there's no correlation. But if you would have had this information on the genetic background, then you can see that there are certain subpopulations that definitely have an increased risk as a result of an environmental exposure. So identification of these subgroups based on mechanistic and relational grounds can be very helpful in these epidemiological studies. So that's why we are trying to get more and more of this molecular understanding of biological processes and more molecular markers in epidemiological study. In the second module, in the second block, in the second module on prevention, I will show some more results of this. And so in the meta-analysis, you can look at certain of these polymorphisms that are very relevant in different types of cancer. Okay, wrapping up, predisposition to cancer. There's only very little pure genetic and very little pure environmentally determined cancer. There's some spontaneous, but most of the cancer causation lies in the interaction between the genes and the environment. So, if you climb the ladder of your DNA, if you try to understand what's going on in the genes, in the modifications, in combinations with environmental exposures, so you actually need not just a DNA ladder, but also insight in your cultural, environmental, peer group pressure, then you may find out where your life is taking you. Okay, that's what I wanted to say in a general introduction on predisposition and particularly on cancer. Are there any questions? Yes.
You have all different kinds of cancers. Does this interaction between gene and environment from all the different cancers that you have to most likely expose to cancer?
Okay, what he's saying is that there are different types of cancer, and does it mean that it's for all different types of cancer the same sort of genes or the same sort of exposure that pull the trigger? Yeah. Is anybody who has some ideas about this? Okay, and there you're saying the percentages may change from one cancer type to another because there may be a little bit more of this gene active in certain, let's say in the liver as compared to the skin. Other suggestions? No, actually that's quite a good answer because some of your gene activity is much more important in the liver as compared to let's say in the kidney because you have different processes. So if you have a specific gene that's polymorphic in an individual, then you can see that certain typical compounds are metabolized in the liver and some other compounds are metabolized in the large intestine. Actually, it was a bit hidden in 2 slides ago, which I skipped. But actually, I had a PhD student on this, so I can talk for the rest of the day on these 2 things. And acetyltransferase 2 is as a slow acetylation, so it's just the second metabolism process of another class of compounds that you see in barbecued meat, the intracyclic aromatic amines. They are related to bladder cancer. So if you have the slow genotype, these metabolites end up in the bladder and can cause cancer. Now, the backside of this story is if you have the high acetylation phenotype, or genotype, sorry, then you have a reduced bladder cancer risk, but an increased risk of colon cancer. But that's because then the first step of the metabolism is taking place in the colon. So indeed, it depends on which, so you cannot say this is a favorable genotype, but only it can mean that your risk of a certain cancer risk factor related to specific tissue changes from one tissue to another. So making it more complex, you can't really say, oh my God, I've got this wrong genotype and now I have an increased risk of bladder cancer. If you would have had the other type, you would have increased risk of colon cancer. So it's what you prefer. Personally, I have no strong opinions about this. Okay, other questions? Thank you for your attention. Okay, one question up there.
If I develop, sorry, if I develop a cancer during my life, and in the meantime I have some sons, do I have the possibility to pass the cancer through by environment and not in my genetic?
And the way I understand it now is that you say, if I develop cancer in early life, can I still pass it on to my children? Well, what do you think? Can you speak up a bit? It's a mutated gene, she says, so you could. It has to happen in your testicles. Now that's a problem for 50% of you, I guess. What he really means to say, if you develop lung cancer, you will not pass it on to your next generation because it's a somatic cell. It's somewhere in your own body. You will not pass it on. If it's what we call a germline mutation, that's not just in your testicles, but for you that might apply, then you can pass it on to the next generation. So if you, actually no, if you smoke or if you work with radiotherapy, if you are assistant of in a radio or oncology treatment, or a dentist, you make X-ray photographs. If you do not use your protection in the right way, then you have indeed increased risk of passing on cancerous to your children. So it has to be a germline mutation, and if it's a somatic mutation in the skin, in your liver, kidney, or wherever, then you won't. It does not mean, however, if you have a family history of lung cancer, and everybody has smoked, that is still somewhere a gene that's a susceptibility gene. So cancer is not a genetic disease in the strict way that if you have this gene you develop cancer, but you can have a susceptibility gene. So, and then it may, if you would know that, if you would sequence your DNA and you know that it's a sort of family history, you can find out if you have that unfavorable gene or gene set. And nowadays we don't talk about one or two genes, we talk about gene profiles, so it's really complex number of genes that you can pass on to the next generation, and it means that you could be at increased risk given the combination of certain environmental exposures. So you could identify that if I have an increased susceptibility, then maybe I should not start smoking, which is in any case something I would advise you. I should drink less. I always say I should not smoke and drink less, that's because I don't smoke and I do drink, so it's reasonable. Don't say I should not start drinking. Okay, further questions? No? Then I will see how we can solve that problem of the Thursday afternoon. And I will